It is my honor to introduce Mitch Guttman. Mitch is a professor of biology in the Division of Biology and Biological Engineer at Caltech. He received a PhD from the Department of Biology at MIT, where he worked with Eric Lander in the Broad Institute of MIT and Harvard, established his lab as an independent fellow in the Broad Institute prior to joining the faculty at Caltech in 2013. He is a recipient of numerous scientific honors and awards, including the NIH Director's Early Independence Award, Pew Stewart Scholar in Cancer Research, Searle Scholar, Ben Barry Early Career Investigator at the Jan Zuckerberg Initiative. He was also an investigation at the Heritage Medical Research Institute, a Robertson investigation at New York Stem Cell Foundation, and the recipient of the Wilson Stone Memorial Award from MD Anderson Cancer Center. Finally, in 2020, he also received the NIH Director's Transformative Research Award. So without further ado, Mitch Guttman.
Thank you so much for the kind introduction and for the opportunity to share some of our work that we've been doing over the last few years. My lab has been really interested in understanding the role of RNA in and particularly non-coding RNAs in shaping nuclear structure and gene regulation. And so many of you may already appreciate that the nucleus is not a random assortment of molecules, but is rather a highly organized spatial arrangement of DNA, RNA, and protein molecules. And over the last many years, there's been a lot of interest in understanding what molecules are present within each of these different nuclear compartments, that is, structures within the nucleus, what drives the spatial and molecular organization of these structures, and what are the functional roles of such nuclear structures in different aspects of nuclear function, including gene expression. And so my lab has been trying to tackle many of these questions. We've developed various different tools to try to study RNA and DNA organization in 3D space, particularly a method that we call SPRITE, which is a split pool-based strategy for mapping interacting RNA and DNA molecules and their spatial arrangements in situ in the nucleus using sequencing-based, using split pool barcoding and sequencing-based deconvolution. And I won't go into detail about how our SPRITE methods work, except to say that using this approach, we can map both DNA, RNA, and RNA interactions, but also where these interactions occur in 3D space. And we can build out spatial models of these interactions using these direct measurements. And so this is an example of a 3D model we can build from our SPRITE data. This highlights on the left certain DNA regions that are organized around a well-known nuclear structure called the nucleolus. And what you can hopefully see on the right is that we can observe these same DNA regions organizing around the same nuclear structure using microscopy-based measurements. The distinction between these is that our SPRITE model allows us to generate this genome-wide across all DNA regions simultaneously versus having to pick out specific sites. And can do this and generate these models at much higher resolution. The model on the left gives us kilobase resolution versus megabase resolution for the image on the right. And this is true more globally. The structures that we can measure and the spatial distances that we can measure using our SPRITE-based measurements show strong correlations with those that we can directly measure by microscopy-based approaches across thousands of different regions. And so using this approach, we've been exploring where different RNAs, and particularly non-coding RNAs, localize in the nucleus. And what we identified a few years back is that when we look at all these different non-coding RNAs, in this image here, about more than 500 different link RNAs that are expressed in mouse embryonic stem cells, that hundreds of different non-coding RNAs localize within precise three-dimensional compartments within the nucleus. And that conversely, the vast majority of the volume of the nucleus is demarcated by a specific RNA compartment. And so this was very exciting to us at the time because it suggested that RNA can potentially play an important role in demarcating different spatial compartments throughout the nucleus. And so to explore what role RNA can actually play in organizing these different compartments, we started to look at what role these RNAs might play in recruitment of different regulatory proteins. And I won't go into all the different examples and details. I'll just highlight one, and this is focusing on an RNA binding protein called SHARP that is responsible for recruitment of the histone deacetylase complex HDAC3. And when you image the localization of SHARP across the nucleus using super resolution microscopy, what you can hopefully appreciate is that SHARP concentrates at specific locations within the nucleus rather than being diffusively localized broadly throughout the nucleus. And if you disrupt the ability for this protein to bind to RNA, what you see is that this compartmentalized structure is now disrupted, with the protein diffusively localizing throughout more broadly throughout the nucleus, suggesting that in fact these discrete RNA compartments are important for guiding this regulatory protein to specific locations and concentrating at specific locations. Zooming into a specific allele and to a specific RNA and specific compartment, what I'll show you is one such example demarcated here with these two that you can see here within the nucleus, demarcated by these two DNA fish loci, alleles, only one of which has the RNA localized within it. And if you zoom in on these two sites, what you can appreciate is that this SHARP compartment, this high concentration of the SHARP protein, only forms over the allele that has this RNA present, but not over the other allele of the same gene when the RNA is not present. And in fact, this is true not just for SHARP, but for many other regulatory proteins, from chromatin proteins, RNA binding proteins, and others, and other regulatory proteins. And the common theme to all these examples and why RNA plays such an important role in acting as such a seed for for driving spatial compartmentalization is because in all of these cases, what these non-coding RNAs can do is that they can form high local concentrations within the nucleus as a function of their accumulation following transcription. And so the process of transcription can create high concentrations of this RNA product. These RNAs, because they're functional as RNA molecules, they can bind to, via their sequence or structure, to different RNA binding proteins, high affinity. They can act to bind different protein complexes, and by doing so, recruit these proteins to this compartmentalized spatial location where the RNA accumulates at high concentration. And in this way can drive compartmentalization of these different proteins within the nucleus. Now, of course, this is not just a feature of non-coding RNAs. Messenger RNAs are also made in the nucleus and can therefore form high local concentrations within the nucleus. The distinction is that messenger RNAs are generally functional through their encoded protein. And so therefore they are generally exported from the nucleus into the cytoplasm where they need to engage ribosomes and be translated into protein. And so even if the protein they encode act in the nucleus, they need to be re-imported into the nucleus where they then have to search through the entire nuclear volume to identify their cognate target sites. And so through the interaction, the binding between a localized RNA and these diffusible protein complexes, RNA can act to seed this compartmentalized and organized structure of proteins within the nucleus. And this plays really important roles across many different functional aspects of gene regulation. And I won't go through all such examples, but I wanted to highlight two very briefly that we've been studying over the last few years, and that is that by seeding such compartments, RNA can act to drive spatial amplification of transcriptional responses, and can act to spatially and kinetically couple biochemical reactions in the nucleus. And the key feature to all of these examples is that by seeding these types of structures, these compartments in the nucleus, you can drive nonlinear responses within these nonlinear responses to um, different stimuli, to these different examples. Okay, so the first example I want to talk about is this example of spatial amplification, and particularly focusing on an RNA and an RNA called exist and a process called X chromosome inactivation. And so this exist RNA is well known to act to silence chromosome-wide expression across one of the two X chromosomes in female mammals. And it does so by localizing across the entire X chromosome, which is about, in the mouse, a little over 167 million base pairs, and acts to silence more than 1,000 different genes. And it does so by recruitment of this sharp, SMART, and HDAC3 complex to each of these sites. Now, the prevailing thinking has been that this RNA acts to localize across each of these different sites and therefore stoichiometrically recruit this repressive complex to each of the genes on the X chromosome. And when you look at it at an ensemble level, this is true. The problem is when you start to think about this at the single cell level, what you realize is that these numbers can't be right. And that's because these numbers can't work. This model can't be right because there are only about 60 to 200 copies of exist within each cell. And so that would mean that you would have one copy of exist that would localize and silence roughly 2 million bases of DNA or more than a dozen different genes simultaneously. And so how can an RNA that can't possibly be localized at all of the genes it will silence act to deterministically silence each of these genes? And put more broadly, how can an RNA that's expressed at much lower levels than its target sites actually work? And so to address this, we start thinking about what is the nature of compartmentalization, and if you think about compartmentalization as by forming a nuclear compartment, you create a smaller volume in which all of these regulatory proteins are now localized. Then even a small decrease in the radius, in the diameter of this compartment relative to the nucleus can lead to a dramatic increase in the local concentration of proteins within that compartment relative to the rest of the nucleus. So for example, if you have a 100-fold reduction in the radius, you would have 100 cubed increase in the effective concentration of molecules within that. And that's because you have volumetric compression. And so if you can concentrate molecules and proteins within such a compartment and drive high local concentrations, we now know that many different proteins can undergo concentration-dependent phase transitions to form condensates. And so by driving these local concentration changes, you can potentially push you can push the concentration regime from the left side of this curve to this right side of this curve to go from a diffusive to a compartmentalized or condensate-like structure. And you can do that in a localized way within the nucleus. And in fact, in the case of the inactive X, this is exactly what happens. I won't show you all the data except to show you this image where you can see that in magenta is where exist is localized. In green is where the inactive X chromosome and where sharp is localized. And you can see that this repressive complex actually accumulates over sites on the X chromosome even when exist is not present. And in fact, this brings us to this broader mechanism where by seeding a high local concentration at specific locations in the nucleus, an RNA can guide and recruit different regulatory proteins to this precise location through stoichiometric binding between the protein and the RNA. And by doing so can can lead to a concentration shift between the concentration of this protein that's present in the nucleoplasm, the broader nucleus, the nucleoplasm, to the concentration within the compartment. And when that concentration achieves a high enough local concentration, it can undergo a concentration-dependent phase transition, where now these proteins can interact with each other, not necessarily and strictly through its interaction with the RNA, but even through these low complexity domains or intrinsically disordered regions within the protein itself to form protein-protein assemblies within this spatially constrained location. And this is what we refer to as amplification, because now the concentration of this protein within this compartment exceeds the concentration of the RNA, that nucleated this process. Okay, I'll go really quickly through the last example. And what I wanted to highlight specifically here is this second feature around spatial and kinetic coupling, and specifically by highlighting that nuclear organization around various different nuclear bodies is a shared feature of RNA processing, from ribosomal RNA processing to siRNA biogenesis to histone mRNAs to even Pol2 transcribed messenger RNAs. And that's associated with the nucleolus, Cajal bodies, histone locus bodies, and nuclear speckles. But what functional role, if any, these nuclear bodies play in RNA processing has been a long-standing mystery, and in part because it's been very hard to disentangle the cause and effect of these processes organizing at these structures versus these structures being important for driving RNA processing. And focusing on nuclear speckles for a moment, nuclear speckles are generally enriched for various different splicing factors. I'm sorry, my animation here is not working well, but they're enriched for various different splicing factors, and that's been known for decades. But speckles were long thought to actually act as warehouses of inactive splicing factors, and that's because when you track where splicing factors are relative to the nascent pre-mRNAs that they act to splice, you actually have activated spliceosomes that actually leave nuclear speckles and travel to nascent pre-mRNAs to mediate splicing. But by looking at these different spatial organization structures using our SPRITE data and other methods that the field has used over the last few years, we've started to learn that, in fact, DNA is not randomly organized in the nucleus, but rather highly transcribed Pol2 genes are preferentially organized on the periphery of nuclear speckles. And so that led us to this hypothesis that perhaps organization of actively Pol2 transcribed genes around speckles can in fact act to increase the splicing efficiency of these highly transcribed genes. And to make a long story short, I will just say that in fact, this is true, but when you look at genes that are organized around nuclear speckles, they tend to have higher concentrations of spliceosomes, they're more efficiently spliced, and that organization around speckles is a dynamic across cell types that leads to dynamic splicing changes. And in fact, simply recruiting a gene to a nuclear speckle, such as through these through an MS2 MCP fusion construct shown here, directly leads to increased splicing efficiency of an otherwise identical mRNA. So simply being close to and spatially organized around a speckle can drive increased splicing efficiency. And so what this example highlights is that the process of active transcription through RNA-Pol2, creating high local concentrations of nascent pre-mRNAs at the DNA site of transcription can drive coalescence of these DNA, of a DNA locus with a nuclear speckle, and by doing so can position it close to nuclear speckles. And by positioning these DNA regions close to nuclear speckles, the concentration of spliceosomes at these nascent pre-mRNAs is higher than they are when a gene is localized farther away from a nuclear speckle. By having higher concentration of spliceosomes, you have higher concentration of these enzymes that can engage and bind to the substrate, the pre-mRNA and the splice sites, to mediate the splicing reaction, leading to higher output of spliced mRNAs for those that are transcribed near nuclear speckles. And my last slide, I just want to highlight that this shared framework that allowed us to integrate and tackle many of these questions, and that hopefully many of you may also find valuable, that are built on this shared split pool-based approach from our RNA-DNA SPRITE methods that allow us to map RNA and DNA spatial organization, our SPIDER-based methods, which I didn't talk about here, but if you're interested, was funded by this TDGC program for mapping, comprehensive mapping of many different RNA binding proteins to RNA, and our ChIP-DIP method that allows us to generate comprehensive maps of hundreds of different proteins, chromatin proteins, histone, TF, transcription factors to DNA. And with that, I also wanted to thank the folks in my lab and the TDCC program and NHGRI and others for funding this and their work in our lab.
I might take you out of your comfort zone into the cytoplasm just for the fun of it.
Sure.
I love all your tools and all your techniques. I'm really happy. I was really happy to see your spider-seq out after I saw it for the first time in Cold Spring Harbor many years ago. So this is more speculation. Do you think, or maybe does your data give you capacity to see this type of kind of a spatial concentration driven by RNA in the cytoplasm? I am thinking about RNA transport, for example, which is something that fascinates me for many different reasons, but I think it's also really relevant for the general community, gene expression. Do you think, do you see anything related to that? Maybe you haven't looked yet, but I would be fascinated to see that.
Yeah, so I will start by saying we haven't actually explored the cytoplasm in great detail, so I'm not going to give you any particularly exciting, groundbreaking results. But our methods absolutely do measure all these interactions in the cytoplasm as well, including RNA compartments and structures and assemblies in the cytoplasm. The primary reason we haven't focused on it is because we're just a nucleus, but also because the mechanism we're working on is a uniquely nuclear mechanism because it is coupled to transcription. But of course, as you're correctly pointing out, there are many RNA granules, assemblies, go by many different names in many different contexts that are cytoplasmic and are functionally really very important, and that can play important roles also in driving condensate formation and both in normal and pathological states in the cytoplasm, and absolutely would be fascinating to use our tools to explore them. Hopefully, maybe something you guys might even be interested in.
Yeah. Okay, there is a question in the chat that I want to address. We have 2 more minutes. So Haryanto to Mitch, are you also looking at silencing mechanism of inactive domain near Speckles, nucleolus of nuclear or nuclear peripheral?
Yeah, I mean, absolutely. We've been very interested in those questions for a long time. We know, for example, that there are RNA-based mechanisms for silencing transcription near nuclear lamina, for example. As is pointed out, right, the nucleolar periphery is often associated with inactive transcription. I don't have any particularly novel insights to provide, except to say that we know that physically being at the nuclear periphery or the nucleolar periphery itself isn't a silencing mechanism. That transcription can occur there and often does occur there. It's that the concentration of many of the different silencing proteins, such as heterochromatin proteins and others, that are also recruited to those locations at the nucleolar periphery and the nuclear lamina that are correlated with and often associated with the transcriptional silencing of genes at those locations.